Our first in the series for 2019 for the history of medicine. It is indeed wonderful to see all of you here and the sustained interest that you've shown in the talks in this series. So as you all know, immunotherapy as a treatment for cancer is a dream that's more than 110 years old. And I thought, who better to present this history to us than our very own Drew Pardoll. So to give you a brief introduction, Dr. Pardoll is the Abeloff Professor of Oncology with joint appointments in several other departments, and he's a world-renowned cancer researcher in the field of immuno-oncology. He has more than 300 papers, more than 25 chapters, and has trained and has spread out indeed his mentees all throughout the world, and there are close to 75 to 100 of them. So he is a well-recognized expert on T-cell immunology and cancer vaccines, and indeed in the field of cancer immunology. So since this talk will not focus too much on his own work, I wanted to highlight some of it here. And so among his many successes, he has identified 3 immune cell types: the gamma delta T cells, the NK T cells, and the innate killer dendritic cells. He discovered PDL2, another ligand for PD1, and described the role of LAG3 in regulatory T cells and its related effect on CD8 effector function. And this has led to a number of, multiple cell-based and antigen-specific vaccines. So one of the really hallmarks of his work is that the basic research always finds its way into the clinic, into clinical translation. So this now he serves as the director of Bloomberg-Kimmel Institute for Cancer Immunotherapy and the co-director of the Cancer Immunology Program at the Sidney Kimmel Comprehensive Cancer Center. So this institute now supports cancer immunology research across many cancer types, including melanoma, colon, pancreatic, urologic, lung, breast, and ovarian cancers. Indeed, a large plate. In these roles, he is leading the pursuit of discoveries that are making it possible to trigger the immune system's natural cancer-fighting abilities. I don't need to mention here the number of places that he's on the scientific advisory board, the number of journals that he is an editor or co-editor of. Indeed, there are many, and these are all the major ones in the field. With that introduction, may I?
Wow, this is intimidating. In our field, it's interesting. I'm not 110 years old. But the reality is that in a lot of ways, and you'll see a little bit of this, our field, while being indeed that old, in many ways has been sort of compressed into the last 10 to 12 years, which has made it very exciting, somewhat breathtaking, and certainly there's a lot more to come. And also, I'm certainly proud that some of the real transformative discoveries, both at the laboratory level and at the translational clinical level, came out of Hopkins, a large team effort, and I'll refer to some of that. So if this is a little bit of a Hopkins-centric history, you'll have to excuse me, but I would at least go so far as to say in this institution that's not totally unreasonable. I myself actually got interested in immunology. I did all of my training here at Hopkins, and I've actually had only one job in my life. They change my title periodically, and every once in a while they give me a cost of living raise, but otherwise I've essentially had one job. My training in science started as an MD, Ph.D. here in 1980. You undoubtedly recognize my two mentors. And at the time we were studying structural and molecular elements of DNA replication in mammalian cells. And we used to do a lot of brainstorming sessions, Bert and Don and myself. And one of the questions that came up was, what is actually the fastest dividing mammalian cell? And it turns out that, at least in the adult, and we were interested in this because of the notion of how does, the complexity of replicating 3 billion base pairs actually figure itself out, and that was involved looking at sort of loop domains on the nuclear matrix. But it turned out that, in fact, the fastest replicating mammalian cell is actually the lymphocyte. When you activate a lymphocyte, it can replicate in as few as 6 hours. And that sort of got me interested in it, but I was supposed to be doing molecular biology, oncogenes were being discovered. Then I went on to the clinic, and I had sort of my own eureka moment about immunology when two things happened simultaneously. The first is, it was when I was on the bone marrow transplant program, and turns out that bone marrow transplantation, even though it was developed purely as a way to rescue patients so that you could give them higher doses of chemotherapy that would normally be bone marrow toxic, it turned out that its anti-tumor effects had nothing to do with that. It all had to do with the contaminating T cells that were transferred in along with the allogeneic bone marrow. And there was this graft versus tumor effect that was really quite powerful. Unfortunately, there was also this graft versus host disease, which was essentially the lymphocytes attacking the normal body. And this is actually a 12-year-old girl, Nicole Churchio, who I got very close to her family because back then the patients spent about 2 months on the inpatient service. So you really got to know the patients very well and their families very well. She got the first unrelated bone marrow transplant and unfortunately died of graft versus host disease, but without any tumor cells. I used to get copies of the various journals sent to my residence box to read at 1 o'clock in the morning to sort of try to keep my science brain going. And right at the time that this was all happening with Nicole, I opened a copy of Nature, and there was a paper by Mark Davis on the isolation of basically the cloning of the gene for the first of the two T-cell receptor chains. And that, in many ways, signified in immunology this inflection point in it going from a very cellular, oftentimes black box phenomenon to something molecular. And from my Ph.D. thesis, I really carried that with me, the notion that if you could really start studying a biological system at the level of genes and molecules in addition to cells, then you could really make some progress. So I decided that that bone marrow transplantation was great, but we should be able to do better than sort of trying to walk this knife edge between graft versus tumor and graft versus host disease, for which actually a lot has subsequently been developed. Actually, a lot of breakthroughs here at Hopkins. So if one thinks about it in first principles, the immune system should be the perfect anti-cancer agent, particularly when you consider the adaptive immune system together with the innate immune system. First of all, the immune system has amazing diversity. If you look at antibody diversity and T-cell receptor diversity, you're talking about 10 to the 15th or whatever, and it's probably a trillion. Each individual's immune system carries a diversity that's about 1 trillion times the sum of all drug libraries of every pharmaceutical company in the world combined. So when you have that level of diversity, you also have specificity. And T cells and antibodies can arguably be 1,000 times more specific than typical drugs. When we talk about drugs for cancer, the off-target toxicities are always an issue. When you talk about antibodies in the treatment of anything, you'll never hear anybody talk about off-target toxicities. You'll hear them talk about on-target toxicities, but never off-target toxicities, because the adaptive immune system is just that specific. And finally, there's weaponry. If you sort of start adding it up, the immune system has at least 16 different mechanisms to basically kill a cell. Now, normally, it's killing virus-infected cells, bacterial, fungal-infected cells, or extracellular bacteria or fungi, but the diversity of weaponry is really great. And finally, one of the most important aspects of the immune system that one might imagine would be very useful for a cancer patient who doesn't want to go through lifelong treatment is, and that, by the way, is a T cell recognizing this tumor cell. These are all other T cells that don't recognize this tumor cell, so they're sort of feeling around, but this is the cell that actually can recognize an antigen in this tumor cell, and it all of a sudden sort of really sticks on, and it's in the process of actually injecting granzyme B through perforin into this tumor cell that will ultimately kill it. But in addition to that, the immune system has memory. This is why our childhood vaccines give us lifelong immunity. So the notion being that if you could somehow tick over the immune system to get a little bit of an upper hand against cancer cells, this would actually be something that the immune system could potentially sustain. So when I came back to Hopkins on the faculty around 1989, 1988, 1989, I was actually in the Department of Medicine, and we sort of started, there really wasn't much in the way of cancer immunology going on, so we started the stealth cancer immunology program actually in the rheumatology division of the Department of Medicine. You may recognize a couple people in this picture. It's been a while. We actually haven't changed that much. Don't laugh too much. And actually, this led to a large effort in developing the first genetically engineered cancer vaccine. And the notion was, at the time, we didn't really understand what the relevant tumor antigens were. So we might as well use the tumor cell itself as the source of antigen. And one of the ways that one could modify immune responses to antigens in that tumor was to engineer it to produce cytokines. Those were some of the early days of cytokines. And people like Steve Rosenberg were injecting cytokines systemically, intravenously, and we're seeing some results in melanoma, but they were also very toxic. And one of the things that we had learned as immunologists is that cytokines are not hormones. They're really not meant to act systemically. They're really meant to act in an autocrine or paracrine fashion within a few cell diameters of the cell producing it. So ultimately, after testing a number of different cytokines in a way that we would take the tumor cells, transfect them with genes encoding these cytokines, and that would essentially be the vaccine. And we actually started with IL-2, but ultimately, as part of a collaboration with Richard Mulligan and Glenn Dranoff at the Whitehead Institute in Baltimore, we actually found that the most potent were vaccines in which we took autologous tumor cells, transfected them with the GM-CSF gene. That became known as GVAX. And that also led really to the appreciation that GM-CSF is a growth factor for dendritic cells, because the way it turned out that this vaccine was working was that GM-CSF attracted dendritic cells, induced them to proliferate. The tumor cells, particularly as a prelude to clinical trials, were in fact irradiated. So they would eventually break up, release their antigens. They would be taken up by dendritic cells, which had proliferated from the GM-CSF that was being produced by those cells before they died. And then these dendritic cells, as we were able to figure out, would traffic to the draining lymph node, and it was there that they would present antigen to tumor cells. This was one of the early examples, and vaccines were developed in pancreas cancer. Liz Jaffe actually grew the very first human pancreatic cancer lines for allogeneic and transfected them, and those actually became the pancreas cancer vaccine. Actually, Jonathan Simons and Bill Nelson, believe it or not, actually made the initial prostate cancer GVAX vaccines and a whole host of others. And there were many vaccine trials between the 1990s and I would say to mid 2000s, even on into the late 2000s. And while there were always these very impressive anecdotal responses, virtually none of them generated in a randomized large phase 3 trial a statistically significant survival advantage, which is ultimately the bar for most cancer drugs to be FDA approved. And it really was in a lot of ways a dark time. And I remember one of the Don Coffey's favorite sayings is, if this is true, what does it imply? Because one of the things that was frustrating is that it was quite possible to show that these vaccines were activating T cells that could recognize the tumor. Ultimately, with various tumor antigens, you could show that T cells were being activated to recognize those antigens, but that all too rarely translated into a regression of the tumor. And so ultimately, the implications there were that the tumor itself was creating a microenvironment that was a barrier to the function of those T cells. And so we had been, the field, was really looking at immune responses in sort of a single vector fashion, that immunization essentially was an accelerator. You needed antigen, you needed so-called co-stimulation, and that gave you an immune response. And once the immune system eliminated the antigen, it was like you took your foot off the accelerator, and the immune system, just like your car, stopped. But like any system that needs to be carefully regulated, in particular that can be dangerous when it's not under control, for every set of accelerators or activators, there are also inhibitors, and that's what needs to be balanced. I use the car analogy all the time, and that's certainly the case. If you were to try to drive your car any distance with an accelerator but without brakes, that would not end well. And so right around the time that we were developing these vaccines, there were basic studies that were going on defining receptors and ligands that were actually inhibitory to immune responses, a lot of focus on T cells, and these are formally called co-inhibitory molecules. And it's interesting because I actually coined the term checkpoint simply for one simple reason, which is that it only had 2 syllables instead of co-inhibitory receptor. And it sort of caught on. Actually, we put it, myself and Jim Allison did an article in the 1990s and used that term. And the cell replication people were really ticked off at us because checkpoints had been really developed as a term by them as cell cycle regulatory checkpoints. But they now, when you say checkpoint, if the person is not in the cell cycling field, they think of these co-inhibitory molecules in the immune system. But I should have actually copyrighted that term. But in any case, could have funded the lab for a long time. So really what the vaccines were just the sort of the accelerator, and it turned out, as I think probably everybody here knows, somewhat ironically, that the real breakthroughs in cancer immunotherapy came from really blockade of checkpoints. This is an older slide that basically is meant to simply show that in addition to recognition of antigen by the T cell receptor, the response that T cells make, which can be very diverse, is regulated by multiple co-stimulatory and co-inhibitory signals. In fact, the first inhibitory signal ever identified, really, that came out of a T cell cloning project, really in the late 1980s, was in fact CTLA-4. And it turned out that, and then the ligands for CTLA-4 were discovered. And then subsequently to that, Tsuku Hanjo discovered PD-1. PD-L1 is arguably the major ligand for PD-1, but we started getting interested in it when we actually stumbled onto PD-L2, which is a second ligand for PD-1, and it turns out to be actually a duplication of PD-L1. When we were actually looking at the chromosomal organization, it turned out that the two genes are actually about 60 kb from each other, and clearly were a duplication. And as it turns out, these really have been the two major checkpoints that have been targeted to this point successfully. I think that probably the next one that's going to be used in conjunction with anti-PD-1 is LAG-3, and that was also discovered actually early on by Qing-Tai Wang, who was actually a graduate student that Chuck Drake and I co-mentored, and LAG-3 came out of that. And that now is actually in phase 3 clinical trials. And we're sort of holding our breath on that. But certainly these were the two beginnings. And CTLA-4 was the first checkpoint. And Jim Allison did a classic set of studies where he showed that if he injected mice with some tumors with a blocking anti-CTLA-4 antibody, that those mice could reject the tumors. And that wasn't new. There were certainly a lot of things that you could do to mice with tumors and get anti-tumor effects. But this clearly represented a new class of immunotherapy. There was a lot of appropriate skepticism about blocking CTLA-4 clinically. And the reason is that in the mid-1990s, actually before the Allison experiments, Tack Mack and Arlene Sharp knocked out CTLA-4 in mice. And as Tack used to say, this is the most dramatic phenotype in a knockout that's not embryonic lethal, that I've ever seen. And essentially, those animals are all dead within 3 weeks of birth. And it's all an immunologic, really a T-cell dependent death. If you eliminate T cells from those animals, they look fine. And basically they die of infiltration, of immune infiltration into virtually all of their organs. So the notion was, this is arguably not the best checkpoint to block because it should cause a lot of autoimmunity. But this went into clinical trials. Actually, a lot of people forget this, but the first demonstration of anti-tumor effects in melanoma were done in the early 2000s that actually Suzanne Topalian and Steve Rosenberg did when Suzanne was down at the NIH. And these are just a couple examples of some of metastases, even a brain metastasis here in a melanoma patient treated with anti-CTLA-4 and a very robust response in this particular patient. But there was indeed a very high rate of immune adverse events, autoimmunity, hyperimmunity. Here's an example of dermatitis. Here's an example of colitis, which is a very, these are actually T cells in the colon. Colitis is one of the biggest challenges in giving anti-CTLA-4 in hepatitis. And in fact, so there was a 12% objective response rate and roughly 23 to 33% grade 3 or 4 autoimmune toxicities. Now, in any other field, if you said, well, I have a drug, its significant toxicities was 3 times the rate of the efficacy, it would be immediately thrown out. But oncologists are used to working with drugs that can create a lot of toxicity and not necessarily that great of an anti-tumor effect. So they pushed on, in fact, this is the randomized clinical trial that led to the first approval of a checkpoint antibody, anti-CTLA-4, called ipilimumab. These two are, so this is a survival curve. These two arms contain the anti-CTLA-4 antibody. This arm is the control chemotherapy arm. And what's interesting is that a lot of clinical trials in chemotherapy measure prolongation in median survival. Namely, how long is it before 50% of patients are dead? And the difference in median survival here was only about 3 months. So that really was not what was impressive about it. What was impressive about it was this so-called tail on the curve, or flattening of the Kaplan-Meier curve. So the idea is if you were alive after 2 years, even though that was only about 20%, you very possibly were cured, but at the very least, you were going to stay alive for years and years and years. And even with further follow-up, this curve has flattened out. The other thing that came out of this was that really because of the toxicities of anti-CTLA-4, the protocol that was developed was only to give it 4 times to patients, every 6 weeks. So basically, patients were treated with 4 doses for half a year, and then they stopped. And despite stopping the therapy around here, in patients that still had residual cancer that the radiologist could say by a CAT scan was still there, nonetheless, you got this plateau. And that was the first suggestion that indeed there was this phenomenon of immune memory, that you could stop your therapeutic intervention and the immune system would sort of take over. This, as you probably all know, led to the awarding of the Nobel Prize to Tasuku Honjo for his discovery of anti-PD-1, and to Jim Allison for those original anti-CTLA-4 experiments. I actually happened to be with him an hour after the prize was announced. The only reason I'm sticking out my tongue here is because I was trying to get myself, Jim, and this is Pam Sharma, his wife, into the selfie. And not being a millennial, I'm not very good at selfies. And so I was really just trying to concentrate to get a decent selfie. Please, nobody put this on Twitter or anything. So why did we get interested in PD-1? Well, beyond just the fact that we got interested in this pathway from having stumbled on to PD-L2. And the reason is that there was a lot of reason to believe that this was a very active pathway in tumors. Because in a lot of tumors, what you see is frequently you'll see a lot of infiltration of lymphocytes. This is staining for CD8. Those T cells are oftentimes in proximity with tumor cells and also macrophages that express PD-L1, and actually now that Janice Taub has developed a good antibody for PD-L2, it's clear that PD-L2 can also be involved. And when you look at these CD8 T cells, oftentimes they will express, the majority of them will express a lot of PD-1. So here you had in the tumor this sort of checkpoint that we could actually visualize pathologically. The other thing is that the PD-1 knockout mice, as in contrast to the CTLA-4 knockout mice, had no autoimmunity at all for the first year of their life. They eventually begin to develop some autoimmune infiltrates, and certainly if you challenge them with an infection, you can see that there is a lot more damage to tissues that that particular infectious organism infects, which basically says that the PD-1 pathway is a counter-regulatory or feedback system to really down-modulate collateral tissue damage, which tumors really look to be co-opting to protect themselves from being killed by T cells. Now, mind you, at this point, this was in around 1990, there was still very little appreciated about the nature of tumor antigens. But with this all together, we hooked up with a small biotech company called Medarex to initiate the very first clinical trials in 2006. This was the original PD-1 team, a relatively small group, and those very first trials were really organized and led by Julie Bramer and Suzanne Topalian. Medarex didn't really have a clinical group, so Hopkins kind of became their translational group. And the very first paper on this very small initial trial came out in Journal of Clinical Oncology. They actually almost rejected it because they said that there weren't enough patients. But they ultimately accepted it, and they didn't really, it really didn't get, at the time, a lot of notice. And it wasn't until the trial was able to be expanded, and that really was able to happen because in 2009, BMS bought Medarex, and they looked at some of the data on the second trial that Suzanne and Julie had launched, and expanded it dramatically, and that led to some presentations at the ASCO meetings in 2012 that were timed to come out concomitantly with a paper in the New England Journal. And I think if you ask a lot of people in the oncology world, sort of when did immunotherapy really get on people's radar screens, they'll tell you it was 2012. But really this started back in the clinical piece started back in 2006, but the science started a lot earlier. And this was just the data from the first set of patients. This is actually a follow-up, but the bottom line was that all of these arrows represent patients with ongoing responses. Now, melanoma has always been considered, human melanoma, a so-called immunogenic tumor. So it wasn't surprising that that there might be responses in melanoma, although quite a few. Renal cancer sometimes as well, considered maybe if there is a second immune responsive tumor. But the most sort of mind-blowing result, and it really had to do with Julie, who was a lung cancer oncologist, pushing us and pushing Medarex to include non-small cell lung cancer in the group. And those responses were really amazing. If you were to ask 100 oncologists in 2010, what were the probabilities that there would be any lung cancer responses to any immunotherapy? 100 of them would say Zippo. And in fact, this is a little bit older and needs to be updated, but this is what I call the chip on the immunotherapist's shoulder slide, comparing the number of patient years annually of tumor regression provided by the now set of antibodies against PD-1 and PD-L1 in all of the cancers for which it has been FDA approved versus the mutation-targeted drugs. And you can see that literally by blocking this one target, there's really quite a dramatic effect. That said, with a few exceptions, which I'll get to, in general, the response rates, at least for single agent antibodies, are relatively low. And so we obviously need to do a lot better, but also, as is with every approach to therapy in oncology, one wants to develop biomarkers that will tell you which patients are going to respond and which patients won't respond. And in that 2012 paper, Suzanne hooked up with Liping Chen and Janice Taub to develop the first immunohistochemistry stain for PD-L1. And in fact, they then, in a blinded way, Janice read them in a blinded way, correlated expression of PD-L1 with clinical response. And what they saw in that small group, and they only looked at tumors in which the biopsies that were available met a certain criterion, so not exactly total real world, but it was proof of principle. And the result was quite astounding, which was that all of the responses were seen in patients that had greater than, by Janice's criteria, 5% PD-L1 positive. And this actually led to PD-L1 staining as the first biomarker to be FDA approved in the use of immunotherapy. Now, the other thing that, that we learned, and this is, I think, a very fundamental tenet in cancer immunotherapy these days, the original view of the increased expression of PD-L1 on tumor cells was that it was constitutive and it was driven by constitutively activated oncogenic pathways. And that is a mechanism in some cases. But in the majority of cases, Janice began to recognize a very simple thing, and which is why actually I personally love looking at slides. It's amazing how much you can learn when you're just looking at slides. She found that just as lymphocyte infiltration into tumors is not homogeneous, neither was PD-L1 expression. And further, there was a lot of concordance between regions where there were a lot of lymphocytes and regions where there was a lot of PD-L1. And this led to this notion of adaptive resistance, which basically says that PD-L1 expression by the tumor is not constitutive, but rather it's adaptive. It is the tumor adapting because it is sensing that it is under threat by lymphocytes that can recognize it and kill it. And the way it senses it is by the cytokines, certain cytokines that T cells make, of which gamma interferon is the predominant cytokine that actually drives PD-L1 expression, not only on the tumor, but also on macrophages. What that says is that, in fact, a lot of patients, we actually believe most patients with cancer, actually do have circulating in their body T cells, at least, that can recognize their tumor selectively. And so the implication is that the old immune surveillance hypothesis that McFarlane Burnett and Lewis Thomas put forward which suggested that one of the natural roles of the immune system was to find and destroy cancers which were arising all the time, and the only time we saw cancer clinically was in those rare cases where the tumor somehow figured out a way to evade the immune response. Just like a chronic viral infection, I mean, 99% of our viral infections, our immune system clears, but occasionally you get a virus that can evade the immune system and it becomes chronic. So what it said is that hypothesis was at least half right, but it also said that tumors can adapt more commonly, more frequently than Burnett and Thomas imagined through these kinds of mechanisms. And this really was the first example of an immunologic adaptation that tumors can make to really resist being killed endogenously. So there are now 14 different cancer types that one of 6 different antibodies against either PD-1 or PD-L1 are approved, and that led to science naming cancer immunotherapy as breakthrough of the year in 2013. This was actually the first time in history that science named not a particular discovery or a particular technology as breakthrough of the year, but literally called out a field as breakthrough of the year. So this was very exciting, and now it's really obviously part of the fabric of cancer treatment. One of the things I learned when I was a fellow was that there are 3 pillars of cancer treatment: surgery, radiotherapy, and chemical therapy. And really now, starting in 2012, immunotherapy has really become the 4th pillar. Now, science called it a breakthrough, and certainly if you follow FDA approvals for cancer drugs, in fact, about 2/3 of FDA approvals for in cancer over the last 2 years have in fact been immunotherapies. So it really does feel like this was a breakthrough that happened in 2012, and this is already totally out of date, but just in showing this acceleration of FDA approvals. But of course, none of these things, particularly clinically, are breakthroughs. All clinical advances are built on the shoulders of basic immunology. And this is just sort of a timeline of milestones in basic and cancer immunology versus clinical immunotherapy milestones. And you can sort of see it. So indeed, Sarah mentioned 110 years old. Coli's toxins were the first therapeutic immunotherapy really given to patients. They were bacterial extracts, basically a form of LPS. They could induce tumor regressions in a handful of patients, but they made patients incredibly sick. In fact, they were called Coli's toxins by New York hospitals. Pathologist Coli was a surgeon at New York Hospital, the precursor to Memorial Sloan Kettering. And Ewing, who was the head of pathology, thought that all Coli was doing was poisoning his patients. And so Ewing actually coined the term Coli's toxins. But it was really starting in the 1970s that some of the real breakthroughs in basic immunology that led to the immunotherapy milestones began happening. The invention of monoclonal antibodies, the elucidation of tumor-associated antigens really to be followed by mutation-associated neoantigens. The elucidation of how T cells recognize antigen, and again, it really wasn't until around 1990 that checkpoints were elucidated. And then it was really only into the 2000s that, so IL-2 was approved for melanoma, though not commonly used. That was in the mid-90s. Tumor infiltrating lymphocytes, really pioneered clinically by Steve Rosenberg, became very fashionable in the early 2000s, but were really not exportable and scalable enough. And so therapy with expanded tumor infiltrating lymphocytes has not really yet caught on. But one of the things that I think really contributed to the explosion in immunotherapy was the success of the so-called chimeric antigen receptor T cells. And I think, did Pat Brown actually give a talk on these? So I won't go into them a lot, but this is, again, something that started back literally in 1989 by an obscure Israeli researcher named Zelig Eshkar, who showed in a very crude way that if you grafted the extracellular region, basically the antigen recognition region of an antibody, onto the cytoplasmic tail of the signaling components of the T cell receptor. The fundamental component actually is the cytoplasmic tail from the signaling molecule that's really part of the complex called zeta, that you could basically target the T cell via the antibody against a cell, but have the T cell kill it like a T cell normally does. So he actually called them T-bodies. They then were renamed actually I'm not sure, as chimeric antigen receptors. And all of a sudden now, about a quarter of a century later, was the first clear demonstration by Carl June and colleagues, and that was actually in 2011. So right at about the time that anti-CTLA-4 was being approved, that the first work on anti-PD-1 had come out, and this was in 2012. So it was really sort of this combination of things. And this is an example of a patient with lymphoma treated with chimeric antigen receptor transfected or transduced or engineered T cells that were adoptively transferred back into the patient. And you can see various big lymph nodes full of lymphoma, and then they basically melt away. And in patients with acute lymphocytic leukemia and B-cell lymphomas, it looks like the cure rate may be on the order of 30 to 50%. And this was actually two versions of this was actually approved last year. So I want to jump back to tumor antigens and some of the more recent advances. And again, a number of these really are Hopkins stories. So again, you'll have to excuse me for being maybe a little bit parochial, but be that as it may. One of the things that revolutionized a lot of cancer genetics was the ability to sequence the whole genome, and in particular the whole exome. The first exome sequences were done here by Bert Vogelstein, Ken Kinsler and colleagues. And as this became faster and faster and cheaper, more of these were done. And what came out of a lot of sequencing of exomes in tumors is that first of all, tumors generally have significantly more mutations than normal cells. But if you look at the different tumor types, there's a range in mutational load or mutational density. In general, melanoma is the highest, and then you have some of the pediatric cancers that are very low. Melanoma can commonly be up to 800 or even 1,000 mutations on average, and then it goes down. When you look at the cancer types that tend to respond at a higher level to anti-PD-1, it's actually, in fact, seems to be skewed towards tumors that have a higher mutational load. And I remember actually after the 2012 paper came out, Luis Diaz running into my office saying, I know why it's melanoma and lung cancer because they have the highest mutational load. My first question was, well, then how do you explain renal cancer? But nonetheless, that seems to be a general case. And if you look at, this was actually an analysis of literature done by Mark Yarchewin and Mark Hopkins and Liz Jaffe, basically plotting the mean mutational load in these various cancers and the probability of response to PD-1 pathway blockade. And you can see there's a pretty impressive straight line. Interestingly, two of the exceptions, one is Merkel cell cancer, and I'll get to that in a minute, and the other, in fact, was renal cancer. Renal cancer, for reasons that we don't totally understand, but may have to do with other genetic defects, have a much higher response relative to their mutational load. But if you go back to this mutational spectrum and look carefully, what you see is that, and it turns out to be the case with all cancers, that there are subsets of cancers that have whoppingly high mutational loads, like from 500 into the many thousands. And it turns out that these are the cancers that actually have defects in their mismatch repair genes. And so they can't repair DNA, so they accumulate a lot of mutations. Actually, Nick Papadopoulos, when he was a postdoctoral fellow with Bert and Ken, actually cloned some of these genes from patients that have a hereditary form called Lynch syndrome, in which they inherit a heterozygous mutation in one of the mismatch repair genes. And when you look at their frequency overall, it tends to be more common in some of the cancers like endometrial cancer, colon cancer, gastric cancer. But overall, when you look at it, it's about 4% of all cancers, and that's about 20,000 cases per year of advanced cancer. So based on some brainstorming and this notion that this cancer type, if it has a lot of mutations, that should have a lot of antigens. It should be like a smorgasbord at a Jewish wedding for the immune system to go after. And so young Lee and Luis actually began a clinical trial. This was sort of one of those amazing eureka clinical trials that doesn't happen too often in one's life. But the follow-up was published in in Science in 2017. The first few patients were published in 2015. And basically what you can see is that this is a waterfall plot. The response rates are about 60% to anti-PD-1. So really very much off the wall. I think it's interesting to just sort of give a little history on, I call this cancer genetics marries cancer immunology. So the origination of the mismatch repair story started in 1966 when Henry Lynch defined two patients with hereditary colon cancer that was different than than familial adenomatous polyposis. It became known as hereditary non-polyposis colon cancer, but again, because that's too many syllables, most people call it Lynch syndrome. That was in 1966. In 1978, basic researchers in bacterial biology defined a set of genes that in bacteria that encoded enzymes that repaired replication defects in E. coli. So these became known as mismatch repair genes. And then in 1993, concomitant with the finding that these mismatch repair genes were mutant in these Lynch syndrome patients, and it turns out that half of mismatch repair defective patients aren't from Lynch families, that they had thousands of mutations. Now, meantime, in a galaxy far, far away, immunologists were discovering immune genes. PD-1 was discovered in 91. It was discovered to be an immune checkpoint not until 1999, and then the first clinical trial in 2010. The eureka moment was in 2012, and that led to this first clinical trial of anti-PD-1, which started in 2013. That was around the time that a Lynch family patient, I think known to a number of people in the audience, named Stephanie Joho, was diagnosed with metastatic colon cancer. She's actually from a Lynch family, and this was her receiving chemotherapy before they finally quit on it because it wasn't working in early 2014. Her sister actually found this clinical trial on the internet, and she entered the trial. This was her in 2016. She's actually 3 years cancer-free. So, the FDA approved, this is actually the first pan-cancer or cancer type agnostic approval for a particular therapy to be given to cancers with a particular genetic mutation. Many, many years after my Ph.D. thesis, but Don Coffey was so excited that the student and the mentor had gotten together. So in 2015, we recreated that picture. I will note that I was the only one who retains their hair. Boy, am I going to get in trouble for this. Remember that, guys. So one final thing, the other tumor-specific antigen besides mutations are viruses, and also Hopkins played a major role in showing that cancer viruses can also be an important target for immunotherapy. This is work that Suzanne Topalian led in collaboration with Paul Niem at University of Washington in Seattle. So it turns out that about 80% of cases of Merkel cell cancer, which is the second most common skin cancer, tends to happen in the elderly are actually caused by integration of a virus called Merkel cancer polyomavirus. It's in the polyomavirus family, and it makes what actually gets integrated is a small constitutively active piece of the polyomavirus's T antigen, and that is what drives the cancer. About 40% develop distant disease, and basically chemotherapy did not work for this. And it's interesting because if you look at the mutational load of patients with virus-negative Merkel cell, those 20%, they even have on average more mutations than melanoma, about 1,200. Whereas the virus-positive versions, even if the pathologists looking at an H&E sample or slice of Merkel cell cannot tell you whether it's a virus-positive Merkel cell or a virus-negative Merkel cell. But the virus-positive Merkel cells, because they have SV40, or not SV40, a homolog of SV40 T antigen driving them, have only on average 10 mutations per genome. Yet, if one looks at their response to anti-PD-1, these are the virus negatives in red, the virus positives in blue, about a 70% response rate to anti-PD-1 in either case. And it turns out that the virus positive Merkel cells, that response is really coming from T cells specific for that very small piece of the T antigen. And this is just a beautiful multispectral imaging picture that Janice took. Basically, these are CD8 T cells that are basically being kept out of this, this cancer by these tumor, the CD8 T cells are in yellow, and there's this one lone T cell that gets in, but there's this gauntlet of PD-L1 that the tumor is turning on and blocking those CD8 T cells from getting through into the tumor. This one T cell isn't going to really do it. You then give anti-PD-1, and this whole gauntlet sort of gets destroyed, and these T cells run into the tumor, and that was kind of a big deal. So I'm going to end there. I'm just going to skip over these. As most of you know, a lot of the Hopkins successes got the attention of a former mayor from New York City who partnered with a former clothier who founded Jones, New York, to help us launch the Bloomberg-Kimmel Institute. It was a lot of fun, a lot of medical students in the rafters, plus a photo bomber, and maybe our next president. And from that little stealth immunotherapy group in 1991 to the PD-1 team in 2006. We've grown a little bit. I have to say that this is about 2 years old. If we were to take a picture now, I think we need a pretty significant wide-angle lens. But it's obviously been very exciting, I think, for all of us in this field to be able to witness really a fairly significant transformation in which 99% of what happened clinically really happened within the last 10 or so years of this 110-year journey, and hopefully we'll keep it going from there. Thank you. Maybe a little over, but maybe there's time for a question or not. I can hang out here Anybody wants to come up?
Sure.
Or-
I'd like to present a small token of our appreciation to Drew.
That's the lunch. That's a free, it's my free lunch.
Oh, no, it's not lunch. It is a very special thing here.
Oh, wow.
Yeah. So
Thank you.
here we go, and may I get a picture of you? Oh, I just fell on it and it's got some nerve damage. It's going to take a long time.
Oh, thank you very much, David.